So as soon as calcium increases and our actin active sites are exposed, we get cross bridges formed and we get our contraction cycle occurring with myosin performing power strokes, pulling actin over itself, shortening the sarcomere, and again across all of our sarcomeres in a myofibril, all the myofibrils in a fiber, and all the fibers in our muscle that leads to the shortening of the muscle and the generation of force. As long as calcium is present in the cytosol of our cell, the actin active site will remain exposed. And as long as the actin active site is exposed, we will have myosin active actin cross bridges being formed and force being generated. And this is critical because we are able, by continually exciting our muscle fibers, to cause calcium in our fibers to be elevated to an extended period of time. And extended here is really brief, but by allowing calcium to be elevated for a second ton of time, we can perform a contraction over a second. If we want to hold that contraction, we are able to hold that contraction by keeping our sarcoplasmic reticulum calcium channels open and keeping intracellular calcium high for a long period of time. And as long as we keep cytosol and calcium high, that will keep those actin active sites exposed, will keep performing myosin actin cross bridges, and our muscle will continue to try to shorten and generate force. All this together, this is our sliding filament theory. The sliding filament theory states that when myosin binds to actin, it's going to pull actin over itself. Those filaments slide over each other, they shorten, and as a sarcomere shorten, force is generated.